Hello, guys, and welcome to another episode of Last Minute a channel that helps you grasp what is going on in the section. Maybe you just started the section in school and you think it's a bit threatening and frightening. This channel helps explain everything to you and make you understand that actually this is not that hard. Also, maybe you've been studying for a long time and now you're tired and you just want to passively learn the night before your exam to make sure you've gotten everything in the bag. This is the channel for you. Today we're still doing a continuation of nucleic acids. I have posted a part one, so it would be very advisable to watch that before you come to this one. But let's get to it. So as mentioned, today we are going to be doing a continuation of nucleic acids, and we're going to be looking at the processes that involve nucleic acids, which will be DNA replication, transcription, translation, and also DNA profiling, which is the importance of nucleic acids in biotechnology. So let's get to it. So what is DNA replication? So DNA replication is just when DNA is making a copy of itself. So this process is very important, especially before mitosis happen, where cells are multiplying or your sex cells are dividing and or forming, which is meiosis. So it's important because you need to make sure that DNA, that your the exact copy of your DNA is everybody got it, everybody understand what needs to be done and what function has to happen in your body. So DNA replication is important with that, making sure that the new cells have the same instructions of what needs to be done in your body. So let's look at the process. So what happens when DNA replication occurs? So the first step is that you have your DNA structure, which is double stranded and is in helix form. So, what happens is the helix will unwind and then it will be in a ladder. It will be just straight like a ladder. And then what will happen is that there are weak hydrogen bonds in between your nitrogenous bases. And then those bonds hold together the strands. What will happen is that those bonds will get broken and then your DNA strand will start unzipping like a zip on your jacket. So they will unzip and then the strands will be broken up and they'll just be individuals. And each strand, each DNA strand will then serve as a template for which a complement DNA strand will be formed. What do they mean by that? Is that, like we had mentioned, that the nitrogen bases have best friends. So each nitrogen base will basically bond with that best friend. So it will make another a strand which will be able to bond, which will have the correct best friends for the nitrogenous bases, if that makes sense. I hope it does. So what will happen is that there will be free nucleotides in the nucleus, and all of those the nucleotides will then build on these strands or each of the strand in attaching to them. The complementary nitrogenous bases, as I mentioned. Adenine will bond with thymine, cytosine will bond with guanine. So those will be building up on each of the strands to form the complementary strand. So this will result in two identical DNA molecules made, each consisting of one original strand and one original and one new strand. So if you zoom in the photo, the picture that I have attached there, you can pause the video and look at the picture. You see the original um, DNA molecule. One strand has A, C, G as the first three nitrogenous bases, and the second strand has T, G, C. So you can see that the strands have the nitrogenous bases that match. So A is matched with T, C is matched with G, G is matched with C. So when they separate, they were complementary to each other and they separated. So obviously when they make another new strand on each other, they will make basically the one that they were with before, the complementary. I don't know if let me make an analogy. So here's an analogy. You have a couple, boyfriend and girlfriend. Boy is so shy, introverted, not that talkative, not like calm. And then you have girlfriend. She compliments this guy because she is very talkative. See, she is extroverted, she is bubbly, she's out there. So they complement each other. They're boyfriend and girlfriend, they're a couple, they complement each other. Then they break up. They break up. The girl's gone her way, the guy's gone his way. So the guy finds a new girlfriend, but the new girlfriend is still complimentary to him. The new girlfriend is funny, extroverted, talkative, energy, and the girl finds a new boyfriend and he is shy, calm, not that talkative, introverted. So they found new partners, but those partners are still complementary to them. So in a way they're still in the same relationship. They have made another a duplicate of the initial relationship that they have. So that is the same thing. That's how DNA replication happens. So I hope that it makes sense. Look at the picture, and you can see when you zoom in that the new strand that the old strand has made is basically the old strand that it was with before. I hope that makes sense. So as I've mentioned, DNA replication is very important. It helps making sure that each new cell has the same information that needs to be passed on, and that's how the process occurs. The DNA molecule unwinds, and then it unzips, and then both of the strands serve as templates to form a new strand, but that is complementary to them to form an identical copy. So you have two identical copies of the original, basically. You have two new strands that are identical to each other and identical to the original molecule. So that's what happens. Hope that makes sense. Moving on, we are now doing transcription. So transcription is a process where DNA is basically providing information on how to make a protein. It is passing on information on how to make a particular protein. And when it passes this information to an RNA molecule forming mRNA, which will be messenger RNA. So it won't make a copy of itself, but it will make a copy of mRNA, which will then, that mRNA will send a message and help make a protein. So here how it goes, the process. So this is how the process goes. So what you have is you'll have again your double-stranded DNA molecule. It will unwind, be in a letter form, and it will unzip like a zipper on your jacket. And in this process, only one strand in the DNA molecule. So the two strands will break up, and the other one is not going to have any function at all. It's just going to be standing there waiting. And one DNA strand will form as a template to form a complementary strand of mRNA. Hope that makes sense. So this mRNA is going to be made. So now once the mRNA is made, this mRNA now has the code for the protein that needs to be made. It has the code of which protein will be made. So this whole process that right now that happened of mRNA unwinding and zipping, having its nitrogenous bases hydrogen bonds broken, exposing its nitrogenous bases, forming a complementary RNA molecule, which is mRNA. All of that is the process transcription. Now the code has been made. Now you have an mRNA strand, which, as I told you, RNA only has a single strand. You have the single strand, so the nitrogenous bases are exposed. So when you look at the strand, and you can separate the strand in threes, basically meaning like when you look at the nitrogenous bases, you take the three, 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 take the nitrogenous bases as a group of three, and So these three adjacent nitrogenous bases on mRNA are known as codons. So each three of these groups of codons or nitrogenous bases, which are called codons, they call on one specific amino acid or they code for one specific amino acid. I'll explain it further when we get to translation. So you'll understand once we get to translation. So that was the process of transcription. Now we are going to look at translation. So when we look at translation, tRNA now is involved. So here's tRNA, as you can see there on the diagram. tRNA has the nitrogenous bases, three nitrogenous bases, as you can see, three adjacent nitrogenous bases. These nitrogenous bases are called anticodons. They code for a specific amino acid. And as you can see, tRNA carries an amino acid. So the anticodons and the codons are going to complement each other. And when they complement each other, that means an amino acid is going to be brought to mRNA, which will allow for the protein to be made because amino acids are building blocks of proteins. So translation is a process where a protein will be made. So mRNA will be read, and with what is on mRNA, amino acids will be called based on what is written on mRNA, and those amino acids are going to form peptide bonds to form a protein at the end. So let's look at the process. So step 1, mRNA is formed in the nucleus. Transcription has occurred. Now mRNA leaves the nucleus and goes to the cytoplasm. It goes straight to the ribosome, attaches to the ribosome, and has its new nitrogenous bases exposed, ready for action. So ribosome or rRNA, ribosomal RNA, helps catalyze the calling of amino acids and the bonding of the amino acids to form the protein. So what happens is that, as you can see in the image there, the mRNA is laying there on rRNA, ready for action. And so rRNA will read mRNA. See, reads the first codons or three adjacent nitrogenous bases on mRNA and say, okay, AGU. So that means I have to call a tRNA or a transfer RNA that consists of anticodons UCA, which will come with an amino acid. That tRNA comes to mRNA, attaches to it, and when it attaches, then rRNA will again catalyze a reaction to have tRNA release that amino acid and leave it there with on the ribosome. So we'll keep calling on tRNA, these different amino acids, calling upon them, and every time tRNA leaves these amino acids, the ribosomal RNA will catalyze the reaction for these amino acids to keep forming bonds, basically holding on to each other until they form polypeptide bonds and they have a protein made. So this is how a protein is made. So the bonds between the amino acids are called peptide bonds. And once there's like a long strand of these amino acids, now a polypeptide. And this reaction of the tRNA releasing an amino acid and leaving it there at the ribosome, and also of the amino acids bonding. It's facilitated or catalyzed by ribosomal RNA or rRNA or the ribosome. And it allows for them to link and link. And when amino acids keep linking, as you know, amino acids are building blocks of RNA, of protein, they form a protein in the end. So that's the whole process of forming a protein. So let's do an analogy. Okay, so here's my analogy. We start from transcription. We have the DNA molecule. The DNA molecule will serve as the cookbook or the recipe, actually. It will serve as the recipe. DNA molecule has this recipe, and then RNA comes and is like, I want to make this protein, I want to make this cake. So it comes in, it attaches to this one strand of the DNA molecule and takes a photocopy, basically. Takes a photocopy or takes a screenshot of the recipe and then leaves the nucleus and goes to the kitchen. What is the kitchen? The ribosome or ribosomal RNA. Goes to the kitchen, goes to the ribosomal RNA, and is like, here is a recipe I found in the nucleus that was given by DNA. DNA had the recipe, and I took it. And now the ribosomal RNA is like, okay, let's make this protein, let's make this cake. So what happens is that our RNA will then go on Uber Eats, start, picking the ingredients, ordering the ingredients. So basically, mRNA has codons, as I mentioned, the three adjacent nitrogenous bases. It looks at this. So one set of codons is gonna code for milk. The next is gonna code for eggs. The next is gonna code for baking powder. So our RNA is like, okay, let me just order this from Uber Eats so that I can make my protein. So order these amino acids or these ingredients to make my protein. Ordering, and then tRNA, which is your Uber Eats or Mr. D or Checker 6060, brings the amino acids. Does it see, okay, it matches with my anticodons. This means I have to take milk. This matches with my anticodons. That means I have to take eggs, baking powder. So tRNA then brings in the amino acids or the ingredients, the milk, the baking powder, the eggs. It brings it to the ribosome in the kitchen. And what happens is the ribosome or ribosomal RNA is then facilitating the mixing, putting together the eggs with the milk and the baking powder, whisking, mixing, putting in the oven and cooking. And then you have your fully formed cake or fully formed protein baked and ready and served. So I hope that makes sense. So transcription is where DNA had the recipe, RNA takes a photocopy of that recipe, and now RNA has a messenger RNA is formed. That's transcription. It leaves the nucleus. This is important. It leaves the nucleus and goes to the kitchen. You can't cook anywhere else besides the kitchen, which is the cytoplasm. Outside the cytoplasm, outside the nucleus to the cytoplasm to the ribosome, which is the kitchen. You go to the kitchen, which is your ribosome, and that's where you start getting your ingredients, which you have your Uber Eats, which is your T or transfer RNA, bringing the amino acids, which are the ingredients. You have ribosomal RNA catalyzing the reaction of these ingredients coming and of these ingredients mixing together and baking and forming your cake, which is your protein. So I know there will be questions where they will ask you to explain these processes. So, if I would advise you when you get your exam paper, take the question paper, put these analogies down. Put your DNA couple breaking up analogy, put your cooking in the kitchen analogy in your question paper, but just before you even look at the questions. And you sit down and then you read through your questions and get ready to answer your questions. I would advise that. So I hope my analogies make sense. I hope they do. Okay, so now let's move on and look at the importance of nucleic acids in biotechnology, in the biotechnology industry. So there's something called DNA profiling. So basically, DNA profiling is using forensic techniques to identify individuals with their DNA. So you look at their DNA, you'll be able to identify individuals by just looking at their DNA. That's DNA profiling. It's a forensic technique. So when they do that, they will create a DNA. Everybody will have, like, they'll take a person, they'll create a DNA profile, which will be a pattern of produced on an X-ray film, which will look like this image. And they'll create this profile and they'll use this profile to compare, to see, to identify, is this person related to you, and all of that. So, as mentioned, why they use DNA profile or why they use the DNA profile for. So DNA profiling is used for paternity testing. Basically, if maybe a man is not really sure if the child that he's raising is his, if maybe the woman had another partner while they were still together and had a baby, and now he's not sure if this is his baby or the other partner's baby. So DNA profile helps with testing paternity. Also, it is used to test for genetic or the probability or causes of genetic defeats. So maybe you are pregnant, you're about to have a baby. When you go to your clinic checkups, they will take your blood and they will ask you if you want to check if your baby has a possibility of having Down syndrome. They will test you and they'll find out if your baby has maybe any gene that shows that okay, they might be born having Down syndrome, so that they can prep for the baby. Maybe if they need surgery once they've been birthed, or anything they need, or for you as a parent to also prepare on how you're going to be able to take care of your baby since they might have Down syndrome. And also, you can also do genetic testing for yourself. Maybe you are considering having kids and you just want to check if there's anything that you might have that is in your family bloodline that you might pass on to your kids without knowing. So maybe you have, maybe in your, for example, like I know that for men, Alzheimer passed down in men. So if your father or your grandfather had it, your father might have it, or not present it but could have it. I think they also can present it and then they can pass it down to you. So maybe you want to just make sure because you heard that your grandfather had Alzheimer, you want to make sure if you have it or not, and if it's going to be a good idea to have kids or how to prepare if you are going to have kids. Also, it is used to establish compatibility for tissue types for organ transplants. If you are receiving an organ, maybe you need a new kidney or heart and you're receiving this heart, they have to check your blood type to see if you guys are compatible and your body won't reject this organ. And also it is used to identify relatives, maybe your loved one passed on in a crime scene away from home with no identification, or they got kidnapped as a kid or something like that, and they now need to identify is this your relative or your loved one, and they take your sample and they sample and compare and see if you are related. Or maybe in a crime scene they find blood, they will take that blood and they will compare it to suspects or previous criminals that are known to see if they might have been involved in that crime. So that is what DNA profiling is used for. So how does it go? So what happens is that to do a DNA profile, what they do is they will take a sample from the people that are going to be made a profile for. They'll either use blood, saliva, hair, skin, or semen. They'll take that, create a profile. They will take that and make sure they're packaging it correctly in safe plastic bags or containers that will ensure that there's no contamination that will occur. And then they will process that sample that they've taken and then they will compare it. Either they have standardized sample there or they'll compare it between relatives or whatever. They'll compare and see if they find any match, and then that's how it works. So we are done. Thank you so much for tuning in and watching this video and clicking on my channel. Please do not forget to subscribe. If you're new here, please subscribe. This channel is only going to get bigger and better, and it's going to help you in every way you need to when it comes to your life sciences. So subscribe right now while you're here, and also share this video with your classmates. Leave a comment down below if maybe there's a part that you would like me to explain a little bit more further, provide you with more analogies. I'll be more than happy to do so. And also sections that you would like me to do in terms of life science in the future, I will appreciate that. And also leave a like if you like this video. And this video you found it very helpful. Like it, please. Again, thank you so much for being here. And I hope you stay. And thank you. Bye.